Welcome to AP Biology. Today I want to talk about oxidative phosphorylation. So oxidative because oxygen is involved, and you phosphorylate ADP to make ATP, which is this part right here. And so last time we talked about the electron transport chain, which is this part. And the electron transport chain takes the electrons from the oxidation of glucose all the way through the Krebs cycle to carbon dioxide. So that gives electrons to these carrier molecules, the delivery molecules, NADH and FADH2. Those give electrons to the electron transport chain. As the electrons pass from molecule to molecule and finally to oxygen, they lose energy. That energy is used to pump hydrogens against their concentration gradient. Those hydrogens, also called protons, sets up a proton gradient. That proton gradient is really important because it's really potential energy. So it's just like a battery where you have a positive side and a negative side. You have a charge separation. And so what you've got here between here and here is a charge separation. Lots of positive protons here and not so many of them here. And so that can be used to make ATP. So this is ATP synthase, which is the molecule we were looking at here. And so this is a better picture of it. And so here's the protons. They're here. There's not so many of them here. That's a proton gradient. That's a difference between here and here. A gradient is a difference. Just like a gradient on a road has a high and a low end, a gradient here means you have lots of something, in this case protons, lots of protons here and not so many of them out here. So it's a proton gradient. Naturally, they're going to go from high concentration to low concentration. So the protons flow through ATP synthase. ASE, it's an enzyme. They go high to low. When they go high to low, that's just passive transport, right? Doesn't take any energy for that. But it is very much like water over a dam. So if you picture water falling down, maybe a little 10, 12, 20-foot drop, and if you picture a water wheel, from if you've ever seen a show from a couple hundred years ago, there might be a water wheel here, and that is going to be able to grab the water and spin. And when it spins, it can turn a gear, which can be used maybe as a sawmill, or maybe that turning, rotating gear can be used to grind up some wheat or something. But that's actually what's happening here. As the protons go through here, it's sort of like water flowing through. It's protons flowing through. And that's used to twist this molecule. And when it twists, it's able to put ADP and P together. It's phosphorylating ADP. Phosphorylating. So it rotates ATP synthase and makes ATP. All right, so how does all that work? Well, oxygen, this wouldn't work if you didn't have oxygen. So oxygen is the final electron acceptor. The oxygen is going to meet up with the electrons that went through the electron transport chain and some hydrogens that are just hanging out in here because they came back through this way, right? Through ATP synthase. And that's going to make water. That's the final waste product. So some vocab words here, the proton-motive force. That's the force of these protons coming out this way. So because there's potential energy here, with these protons all being stuck up here, that's really a force, and we call it the proton-motive force. It's protons or hydrogen ions move with a concentration gradient. It's also an electrical gradient because you have positives here and not so many here. That releases potential energy, which is then used to make ATP. Okay, so it's sort of like water, you're going downhill. Because oxygen is needed to phosphorylate the ADP to make ATP, we're going to call it oxidative phosphorylation. You need to know the difference between this and substrate level phosphorylation. So substrate level phosphorylation is just an enzyme grabbing a substrate, taking a phosphate from it, and making ATP. Oxidative phosphorylation is a lot more involved, right? You need to oxidize glucose to put the electrons on these carriers. The carriers have to come over here to the electron transport chain. The electron transport chain has to make a proton gradient. which is a difference between the intermembrane space and the matrix. It's a difference, it's a gradient in protons. Then the protons go, that's potential energy, right? Just like a battery, right? The protons being all stuck here. When they come out through ATP synthase and make ATP, they help this enzyme phosphorylate ADP to make ATP. You need oxygen for all that, right? Because the oxygen pulls the electrons off of this guy. If oxygen didn't pull the electrons from here, they would back up. You wouldn't have electrons moved from guy to guy to guy. You wouldn't be able to put the protons in here. You wouldn't be able to make a proton gradient. They wouldn't be able to come out this way and make ATP. So you need oxygen for all this. So we call it oxidative phosphorylation, much more involved. Another vocab word, chemiosmosis. I personally think it's kind of a stupid name because it has nothing to do with water. But the guy who figured this out was like, wow, these protons moving through here looks just like osmosis because they're moving. It's facilitated diffusion. And so they call it chemiosmosis. All it means is the protons moving out through ATP synthase. So the buildup, so this is supposed to be an image of the electron transport chain, really, really simplified without much of a chain here, huh? But that makes these protons. So the buildup of the proton gradient by the electron transport chain is made so you can have protons that will then flow through ATP synthase to make ATP. So what connects the electron transport chain to ATP synthase? Well, the thing that connects them is the proton gradient. So that's the key here. The proton gradient links the electron transport chain to ATP synthesis. AP wants you to know that for sure. So the thing that connects electron transport chain and ATP synthesis is this proton gradient. If you poked holes in here, right, you have a little hole here. If you poke holes through here, you don't make any ATP, or at least you make way less. Okay, so during oxidative phosphorylation, chemiosmosis couples the electron transport chain to ATP synthesis. So here's the electron transport chain. It's this stuff. Chemiosmosis is hydrogen ions going through ATP synthesis to make ATP synthase to make ATP. And then oxidative phosphorylation is both. It's the proton gradient that connects the two of them together. So energy coupling, this one is an energy releasing or an exergonic reaction. This one is an energy requiring or an endergonic reaction. Every reaction that requires energy uses an energy releasing reaction for the energy in our bodies. So in this case, the endergonic reaction is making ATP, so ATP synthesis. How do you get the energy from that? You get it from the proton gradient. So protons moving out of, or out through ATP synthase. Notice that the amount of energy here, well, it's actually this amount of energy is less than this amount of energy. So you definitely have a little bit lost each time. So this would have to be lower. Okay, so the energy stored in the proton gradient across the membrane couples the redox reactions of the electron transport chain to ATP synthesis. So bottom line here is that you are oxidizing glucose in little steps, and you're able to use that energy to make ATP. This is a picture of the amount of energy that you have in each of these molecules. So NADH has electrons with a lot of energy. Those electrons lose a little energy when they're picked up by this first carrier, then the next carrier, and the next carrier. You keep losing a little bit of energy through each of these, and all the way down to here. So you start at 40 kilocalories per mole for the electrons here, and you're down to about 20 by the time you get to the end. Oxygen, by the time you get there, the electrons have very little energy. So each carrier is more electronegative, which means they're holding the electrons tighter to themselves. So this one is more electronegative, more En for electronegative, than like this one, right? This one is less electronegative. So if you're more electronegative, you hold the electrons in tighter, which means they're at a lower energy level, or at least tighter, closer to the nucleus, which means they actually have less energy. So less energy here in those electrons. It's all controlled oxidation. The energy is released and it's used to make ATP instead of combustion. This is just the idea that you can have more than one electron transport chain in mitochondria. In fact, you have lots and lots of them. Every single little cristae has, a bunch of them, right? So this is your inner mitochondrial membrane. Protons will be out here, right? Okay, this is just a visual for you to see the whole thing all together. Glycolysis to pyruvate, here's the Krebs cycle. That breaks down all your glucose to CO2. The first step here is that electrons are harvested and carried to the electron transport system. They're carried by these delivery molecules, NAD and FADH2. The electrons then move through the carrier molecules, so that's step 2. The electrons provide energy to pump these protons against their concentration gradient. Step 2 is active transport, or proton pumps. Right, they're proton pumps. Even though they don't require ATP, they require energy from the electrons. Step 3, oxygen joins the protons right here, and some electron. So oxygen takes the electrons from here, joins some protons, makes water. Step 4, you've got this proton gradient. As the protons come back through here, that's the energy needed to make ATP. So there's potential energy in the gradient. And as they come through, that's used to make ATP, chemical potential energy. As they come through here, that's just facilitated diffusion, which is a type of passive transport. It's a little bit tricky here because passive transport doesn't require energy, has nothing to do with ATP, but here it is making ATP. Kind of wacky, right? A little bit wacky. All right, so you get much more ATP made by oxidative phosphorylation than substrate level phosphorylation. Substrate level phosphorylation is just when you have a phosphate and you have an enzyme that pulls it off and puts it onto ADP. So in glycolysis, you make 2 ATP net, and in Krebs, you make 2. Those red circles are from substrate level phosphorylation. In the Krebs cycle, you make 6 NADH, so 6 times, that accounts for 3 protons getting pumped, because remember, go back a little bit. Okay, so here's NADH pumping, giving the electrons, right, to the electron transport chain. That is responsible for pumping 3 protons through, which is responsible for making 3 ATPs. So you multiply 6 times 3, right? That makes 18 ATP. That's the oxidative phosphorylation is what I'm doing in blue, or kind of looks green. Let's see, here's NADH, 2 times 3 is 6. Here's 2 NADH, 2 times 3 is 6, but it takes some energy to get these folks into the mitochondria. So that's only going to be 4 ATP. And then FADH2 only pumps in 2 protons instead of 3, and so 2 times 2 is 4. So anyway, if you add all this up, I think that's 32 ATP. Whoops. 32 ATP by oxidative phosphorylation, and by substrate level phosphorylation, you're only making 4 ATP. So altogether it's 36. Whoops. Altogether it's 36 ATP, but you make much, much more with oxidative phosphorylation, which is all like the electron transport chain and all that stuff. Okay, AP claims that you don't need to know any of these names of molecules or intermediates or whatever. That's what they're saying here, but I have absolutely seen cytochrome on AP tests. So make sure you know the term cytochrome and that it has to do with membrane proteins, parts of the electron transport chain, whether it's on a mitochondrion or a chloroplast or whatever. Cytochrome is a word that I want you to know. Whoops, cytochrome. Okay. So what happens if oxygen is unavailable to organisms that do aerobic cellular respiration? Well, if you don't have oxygen, then electrons aren't passed along the electron transport chain, which means you're not making a proton gradient which means you have no protons to go through ATP synthase, which means you're not making ATP, which means you have no energy and you don't last too long when that happens. So the electron transport chain backs up. Nothing can pull the electrons down the chain. FADH and NADH2 can't even unload their hydrogens, so you can't even do glycolysis. ATP production stops and you run out of energy and life stops. It's a bad day.